Vitamin D has one of the most paradoxical histories in modern medicine. It began as a life-saving discovery, curing rickets and transforming pediatric health, only to later become one of the most feared and misunderstood therapies in clinical practice. For decades, vitamin D was reduced to a narrow role in bone health and calcium metabolism, largely because early high-dose experiments raised concerns about toxicity. But today, with advances in immunology, molecular biology, and microbiome science, we now understand something essential. Those early researchers were not wrong. Their conclusions were simply incomplete. In the early 20th century, vitamin D was identified as an anti-rickettic factor. Its clinical effects were dramatic, almost miraculous. As a result, enthusiasm exploded. Physicians began administering extremely high doses, often hundreds of thousands or even millions of international units per day, using crude and poorly standardized preparations. At that time, vitamin D was not recognized as a hormone. There were no assays for vitamin D metabolites. Parathyroid hormone could not be measured. Magnesium and vitamin K were unknown as cofactors, and calcium regulation was poorly understood. Vitamin D was viewed almost exclusively through the lens of calcium. That conceptual limitation would define the next phase of its history. In 1937, Steck and colleagues published their landmark work on what they termed vitamin D intoxication. They described hypercalcemia, renal stress, and systemic symptoms in patients receiving massive, uncontrolled doses. But here is what history often forgets. This was not a study designed to invalidate vitamin D therapy. It was a toxicity characterization study conducted during an era of unregulated dosing, wildly variable preparations, no cofactors, no monitoring, and no understanding of individual biological variability. What Steck demonstrated was calcemic toxicity under chaotic conditions, not the failure of vitamin D as a therapeutic agent. A few years later, Charles Slocum evaluated vitamin D in chronic inflammatory arthritis. His work is frequently cited as evidence that vitamin D does not work. But when we read the data carefully, a different picture emerges. Between 50 and 68% of patients improved. Many improved without hypercalcemia. Renal changes were reversible, and responses varied dramatically between individuals. Slocum's real conclusion was not failure, it was variability. In modern terms, he unknowingly described heterogeneous vitamin D receptor responsiveness, without having the biological framework to explain it. Instead of refining understanding, medicine retreated. From the 1950s through the 1980s, vitamin D was reduced to a bone nutrient. High-dose use was stigmatized. Regulatory limits became extremely conservative. Immune, neurologic, and endocrine effects were ignored. This was not evidence-based caution. It was conceptual oversimplification. Vitamin D as a steroid hormone. Everything changed at the end of the 20th century. Vitamin D receptors were discovered throughout the body. In immune cells, in the brain, and in nearly every organ system. Vitamin D was no longer just a vitamin. It was recognized as a pleiotropic steroid hormone, regulating innate immunity, adaptive immune balance, neuroimmune signaling, mitochondrial function, and epigenetic expression. But as this understanding expanded, a new paradox emerged. The missing piece, vitamin D receptor resistance. Despite adequate, and sometimes very high, blood levels of vitamin D, many patients failed to improve, developed autoimmune disease, experienced neuropsychiatric disorders, or remained chronically inflamed. This contradiction is explained by vitamin D receptor resistance. In the modern world, VDR resistance is driven by ultra-processed diets, loss of microbial diversity, intestinal hyperpermeability, endotoxin translocation, chronic systemic inflammation, autoimmune and neuroinflammatory signaling. This inflammatory terrain interferes with VDR expression, nuclear activation of the receptor, receptor binding and transcription, and downstream gene signaling. In other words, the message is present, but the receiver is offline. Neuroinflammation and the gut-brain axis. This same mechanism explains the rise of conditions such as ADHD, autism spectrum disorders, depression, schizophrenia, Parkinson's disease, Alzheimer's disease. These conditions share common features: gut-brain axis disruption, microglial activation, cytokine-driven inflammation, impaired steroid hormone signaling. Vitamin D deficiency is often secondary. VDR dysfunction is the amplifier. The Coimbra Protocol represented a major evolution. It recognized that autoimmune disease requires higher vitamin D exposure. Calcium metabolism must be tightly controlled. Parathyroid hormone suppression is a functional marker. Monitoring is essential. This restored safety and legitimacy to high-dose vitamin D therapy. But one question remained unanswered. Why do some patients still fail to respond, even when dosing and monitoring are correct? The LGS Protocol. VDR renewal through anti-inflammatory diet. The LGS Protocol represents the next and most comprehensive step established by Dr. Eduardo Beltran. It does not begin with vitamin D dosing. It begins with vitamin D receptor renewal. Its core pillars include microbiome restoration, intestinal barrier repair, anti-inflammatory dietary signaling, individualized, cofactor-supported vitamin D use. Vitamin D is no longer treated as a blunt tool. It is used as a biological signal, introduced only when the system is capable of listening. This resolves the Steck-Slocum paradox. What early researchers observed as inconsistency, transient response, and toxicity, we now understand as unaddressed receptor resistance and gut immune dysfunction. From dose-centered to receptor-centered medicine, the history of vitamin D is not a story of failure. It is a story of premature conclusions drawn from incomplete biology. Early researchers identified real risks, but lacked the tools to interpret them. Mid-century medicine retreated instead of evolving. The Coimbra Protocol restored safety. The LGS Protocol completes the arc. It shifts medicine from asking, how much vitamin D should we give, to asking, is the body capable of receiving the signal? This is not just about vitamin D. It represents the future of medicine, a receptor-centered era where disease is understood as a failure of biological communication, not merely a lack of molecules. Vitamin D does not heal in isolation. It heals when the terrain allows the receptor to listen, and that terrain, in autoimmunity, neuroinflammation, and chronic disease, begins in the gut.